travel to get to the inner circular innermost oblique. It needs to get into the blood to do that. So it's absorbed into the capillaries of the mucosa, gets out to the bloodstream, comes back to the stomach, and activates smooth muscle and activates our parietal cells. It's going to do both the motility and the enzyme release, similar to the vagus. So we're going to see this pattern both in the stomach and the small intestine, where we have nerve activity and hormone activity regulating up, increasing activity and decreasing activity. So now we also have inhibitory activity. The stomach needs to be given time to do this mechanical digestion. When we get to the small intestine, that's going to be involved in that. So, cephalic. All right, sometimes the thought of certain food or just the thought of any food can make you sick. When I was in high school, for some reason, I have no idea, my two sisters and I decided to throw my parents a dinner party. We went so far as to, my sister created a 3-foot sculpture of paper mache of a Italian type image of statue of a woman carrying a water vase, big jar over her shoulder. And it was a very simple dinner, Italian, spaghetti, that kind of thing. The day of the dinner, we all got the flu. And we went ahead, we had this tiny little room about 10 feet by 12 that was our music room, where the piano and one chair was. And we set up a card table with a fancy tablecloth and china. But we were so sick that we just kind of laid around the floor while our parents sat at the table and ate dinner. And for a year, the smell of tomato sauce made me nauseated. All right, so that's the cephalic phase, where either a bad smelling food, anybody ever heard of durian?
Yes.
Okay. That's a fruit in Asia, in the Philippines, that I have never gotten past my nose to be able to taste it, but my dad loves it. He had come back from islands one of the southern islands in the Philippines, and had purchased some durian and had it sealed in a glass mason jar in his suitcase, but apparently the smell still permeated throughout the plane. And it's the kind that just makes you kind of gag. We ran out of the house when we brought it into the house. We couldn't even stay in our bedrooms. And so everybody in the flight, and the stewardesses were trying to go through, everybody's, who has the durian? Well, they didn't think this American guy would be eating durian, so they didn't even check his luggage, and that's where it was.
Wow.
What
are they going to do? Reject it?
But so some foods can smell so bad, certain cheeses and stuff, that you can't get it past your nose, all right? Or you can have a memory of something that affects your smell. That's still cephalic, all right? Then we have the sympathetic nervous system, which is opposite to parasympathetic, so that's going to decrease activity. And then we'll talk about small intestine when we get to the small intestine, but obviously, as we mentioned before, when we talk about the pyloric sphincter, as acidic food empties into the small intestine, it needs time to mix the bile salts and to mix the pancreatic enzymes and to mix the pancreatic bicarbonate ions. So we want to decrease the motility of the stomach to provide time for that to occur in the duodenum, and so we have a reflex activity from the small intestine that provides that both through hormones and through DERs. Okay. So this, by the way, well, I'll mention that in just a moment. It's your extra credit for your exam. So this is showing, it's a little blurry because I enlarged the picture a bit, but this is just showing in a schematic. If all those words throw you off and your brain shuts down when you see all those words, here's a schematic of that where we have increased activity from vagus nerve. So the red is stimulation, increasing activity of these cells, and the hormones, the G cells, also increase the activity. The next couple of slides are just showing the cephalic, gastric, and intestinal phases of what I just went through. Now, when we get to the small intestine, I'm going to spend more time on this diagram. And this information is your extra credit on the digestive exam. Okay? Again, I don't trust my interpretation of your drawings. So I will have the diagram on the board, on the paper, and you will fill in, I'll give you an example, you will fill in various molecules or nerves and tell me where it goes to. So in other words, gastrin would be going to the stomach, and you will tell me with a plus or a minus sign, does it stimulate or inhibit activity at that particular organ. Okay. So rather than a table, a pictorial kind of aspect of it. All right, so now we've reached the most important organ where the both intermediate and, most importantly, final step events occur. Notice the pH, 8 to 9. So as Joe said, we're back up in an alkaline activity, and this is due to not the small intestine, but pancreatic activity, the bicarbonate ions of the pancreas. We do digestion of all of the carbohydrates, all proteins, and lipids, because by the time we get to the large intestine, that activity is complete. So both digestion and absorption of these components are going to take place. Now on Tuesday, I provide you a diagram of the 3 different regions of the small intestine and what happened. In each, here we're going to stop a bit and look at that curvature, which is the duodenum, and spend a little bit more time on the intestinal phase of digestion. I don't really need to write liver up here anymore, do I? You guys know that. I realize my liver's a little small in comparison, but all right, so talking about the stomach, looking back at our gastric phase, we have food in the stomach. So is that going to be excitatory or inhibitory to enzyme release and motility? Excitatory, all right? Vagus nerve is going to go to the stomach. It will go to the pancreas and to the liver and where have it going to the pancreas? To the small intestine. Is that going to be excitatory or inhibitory?
Excitatory.
Excitatory. Notice it is also going to our glands. The vagus nerve has receptors for acetylcholine in the liver as well as in the pancreas. Okay. Sympathetic nerves, and here we're talking about the greater splanchnic that's descending down from the thoracic cavity. And that's going to go, let me draw that in a different color. Well, same thing. I'll draw that as a dotted line. Is that going to be excitatory or inhibitory?
Inhibitory?
Inhibitory, okay? Now, at the pancreas, it is inhibitory to insulin, but it's going to do glucagon, all right? So that part we'll talk about on Monday when we talk about Tuesday next week when we talk about hormones. So that's kind of summarizing a bit of the vagus nerve, okay? Now we have acidic chyme. So we go through here. So we have, go back to the previous slide. So we have acidic chyme, and remember chyme is what we call the churning material that is leaving the stomach. Anybody remember what we call the chewed food that we swallow? The bolus, okay. A bolus is a broad term. It's used when you give a bolus of medication, so you give a large amount first and you follow it with something else. So it means more in others, it means something different in other situations, but in this realm of context, it refers to what you're swallowing, the mouthful that you're swallowing, okay? So now we have the food is passing through the pyloric sphincter that you see right here. Acidic chyme passes into the duodenum. So now we have, let's see, let me add one more thing up here. Gastrin, which is our hormone, okay, and that is primarily going to go to the stomach and it's excitatory. It does also affect the intestines, but focusing primarily on the stomach here. So when the acidity enters the duodenum, we had enteroendocrine cells in the intestinal gland. Right? At the base of the glands, just like at the base of the gastric glands. So these are going to release several hormones. Secretin and cholecystokinin. Actually, let me write this hormone in blue so I can be consistent here. Bless you. So the duodenum, in response to acidic chyme, is going to release these hormones. So our stimulus is going to be acidic chyme and secretin, which is a hormone. Cholecystokinin, which is a hormone, they're going to get into the blood, that's their hormones, and via the circulatory system, they are going to do a variety of activities, all right? So, secretin going to the stomach is going to decrease activity. It's going to be inhibitory to the stomach. It's going to slow it down so that, again, the small intestine has time to deal with all the material that's coming into the small intestine. Cholecystokinin is also going to bind to cells, and I'll just piggyback it onto the same line here so we don't get too many lines. Cholecystokinin is also going to be inhibitory to stomach motility and stomach secretions. Okay? Now, secretin is going to be primarily to response to low pH, whereas cholecystokinin is going to be primarily to increase in lipids. But anytime we have food coming from the stomach into the small intestine, it's going to have a low pH. Considering that secretin is released primarily in response to low pH, what other organ do you think it would be significant in affecting the activity of?
Pancreas.
The pancreas, all right? So secretin is going to go to the pancreas and be excitatory in the release of insulin. Whoops, I didn't want to talk about that just yet. Excitatory in the release of bicarbonate ions. Because that's going to raise our pH. Okay. Cholecystokinin, being those receptors being sensitive to the release of, to the presence of bile, is going to bind to receptors in the gallbladder. Now, does the gallbladder synthesize bile salts? Bile?
It stores it.
Correct, it stores it. So we have this thin-walled sac of bile fluid containing bile salts. So in response to cholecystokinin, the gallbladder will contract and send bile with its bile salts down through the common bile duct in the ampulla of Vater and into the duodenum. Okay? The vagus nerve does that too, okay? But not as strongly as having cholecystokinin. What is the organ that makes bile? The liver. So secretin, interestingly enough, binds to cells in the liver and is stimulatory to the actual synthesis of more bile. So with secretin, you're going to see more bile flowing through the bile canaliculi, whereas cholecystokinin is primarily going to be the contraction of the gallbladder and what's already stored.
And so acetylcholine is what activates those two components, or secretin in the first place?
Acidic, the food coming into the stomach is always acidic. If it also contains lipids, you'll see an increase in the cholecystokinin release than if it didn't contain lipids. Okay, but it's always going to be acidic, and so secretin is always going to be a component of that response. All right, now what was the other component of the pancreatic ducts besides bicarbonate ions?
Pancreatic.
Pancreatic enzymes. So that's a role of cholecystokinin.
It's everywhere.
I was like, it does heck of stuff. My diagram looks ugly now.
Okay. Which is why I don't let you guys draw for me, because I'd be spending an hour in each drawing. So this is going to, cholecystokinin is going to be active for increasing the ACE and R synthesis, release of the digestive enzymes. Now, don't memorize this, okay? How many times did I say that to you in the last? I'm almost done with my semester with you guys, after a whole year. So, cholecystokinin, lipids, what's an enzyme from the pancreas? Pancreatic lipase. So if you think of that in sequence, it makes sense that cholecystokinin, rather than secretin, is going to do the enzymes if it's involved in lipid breakdown anyway with the bile salts. Alrighty? So as we break down our polysaccharides to disaccharides and disaccharides to glucose, elevated glucose in the blood is going to be excitatory to the pancreas making insulin. Okay?
Now we see the part we understand.
So it's a cause and effect. So after break, I'll give you a quick peek, no picture taking. Have to turn off your camera. A quick peek of the diagram I'm talking about. Okay? Just because I always spend 15 minutes trying to explain it to you during the exam. Not because the information is complicated, but try to get you guys to understand what I want you to do. Essentially, maybe I will do the quick peek and maybe I won't. But essentially, what I will have is a box. So I would have cause and effect. Anything that has a box beside it is, you have to tell me whether it's excitatory or inhibitory, okay? So up here I might have a list of food in the stomach. Food in the stomach is not going to facilitate the release of cholecystokinin. However, if I have chyme in the duodenum, that will. So you'll draw a line to that box, and you will put a plus or a minus if it causes that to increase. So if chyme causes a decrease in cholecystokinin, you would put a minus. Okay? If chyme causes an increase in cholecystokinin, you would put a plus. Okay? Now, cholecystokinin itself goes to the gallbladder. Okay? So then you would draw a line from cholecystokinin to the gallbladder and put a plus. Cholecystokinin goes to the stomach, so you would draw a line from cholecystokinin to the stomach and put a minus sign, because it's inhibitory to the stomach. So that's kind of, rather than just filling in a chart, that's kind of, you will be drawing in lines from what, like glucose in the blood, you would be drawing a line to the pancreas and putting a plus for insulin. Okay. So you're going to draw lines from the causative effects to whatever they're causing to happen, put a plus if it increases that amount, or a negative if it decreases that. And that's essentially what that chart is. Okay? So now that we have food is moving in here, so this is a summary of what we just got through saying. All right, so the release of cholecystokinin and secretin. Cholecystokinin and secretin go into the bloodstream. Because of cholecystokinin, the gallbladder will contract and release bile. Because of secretin, the liver will start to make more bile. Because of cholecystokinin and secretin, the stomach will slow down and give a chance for the digestive enzymes from the pancreas and the bicarbonate ions from the pancreas to be mixed in the duodenum. So this is in the PowerPoints for lecture, and you can kind of summarize that best, draw your own. Okay? So either write out what's happening or write out what's happening and then relate that to a picture, kind of a flow chart, a concept mapping type of thing is really what we're working with here. Any questions on that previous material? Yes.
So at the beginning, you have food coming into the stomach and it's excitatory on the vagus nerve?
And the G protein, the G is the gastrin.
Okay,
So it's going to excite the vagus nerve and it's going to cause the release of gastrin.
okay,
And so then we start to release hydrochloric acid and pepsin, gastric lipase. And the gallbladder will start to, mildly, it's not a strong stimulus to the gallbladder. That's the gastric phase part.
Does gastrin, is that what makes you feel full?
No, gastrin is a excitatory hormone to the stomach activity. So like, we do the same thing vagus does, but on instead of via acetylcholine receptors, via gastrin receptors.
Okay.
The hormone that makes like CKK or something.
CCK?
CCK or CKK, like that.
The CCK is the cholecystokinin that slows down the stomach.
Okay.
So stretch receptors, as you put more food into the stomach, that will cause the hypothalamus to cause you to feel full. All right, let me just see if I want to do let's just take a short break here, and then we'll look at start digestion. So 2-minute break. It'll be a good place to stop.